see an accumulation of MCM molecules throughout the slide, whereas in its absence, you basically see a few smudges of probably denatured protein that are sort of hanging out on the surface of the slide. Now, to show you this control, I'll tell you that not only does this control work with UV.org or Origin DNA, or we prevent HB hydrolysis, all of them inhibit this, and these are all hallmarks or characteristics of helicase loading that's been determined in other types of biochemical assays as well as in vivo. So, we had an assay, how do we use it? So the first thing I want to address is the stoichiometry between the Origin DNA, helicase loading proteins. So to do this, we took advantage of the ability to label two different proteins and monitor the DNA at the same time. And we focused initially on CDC6 and CDG1. And the reason for that is that we knew CDC6 and CDG1 were essential for MCM loading. Okay? However, in gene-based assays, the association of these proteins was essentially not detected unless we inhibited HB hydrolysis and stopped the reaction at this post-CCM step of the reaction. Otherwise, it was as if they were never there. We knew they had to be there because they were essential for the reaction. So we thought the single molecule setting would allow us to monitor these events and get an idea, for example, how many CDC6 molecules does it take to load two MCM heteros. So we did this again by labeling either CDG1 and MCMs or CDC6 and MCMs with different colored fluorophores so we can monitor both molecules at the same time. I'll first show you the example with CDG1 and MCMs labeled, and then I'll show you the example with CDC6. So I've shown in this left-hand panel, you can see traces of CDG1 association with DNA in green and MCM association with DNA in red. And we've shifted the baseline of the MCM trace up so you can see both traces simultaneously in the plot. The first thing I hope you notice is that the MCMs associated with the DNA in a one-at-a-time fashion. We see an increase of fluorescence here, and then a second increase, the second arrow illustrates. Now, this could be because we have two MCMs loading in the first step and another two in the second step. However, we can eliminate this model and demonstrate that it's actually one MCM and then a second MCM using a process called photobleaching, which stochastically inactivates fluorophores. So we can take, at the end of the reaction, we can turn up the laser so that we see photobleaching, and this is because it's a stochastic process. If there's two fluorophores, the fluorescence will decrease in stepwise fashion in 2 steps. If there are 4 fluorophores, we see 4 steps. This means that in many of these experiments, and we always see a 2-step loss of fluorescence, indicating that this is a first and a second MCM protein. The other thing I think you'll clearly see is the time of CDG1 association is identical to the time of the MCM association. However, CDG1 comes off rapidly, leaving behind the MCM molecule. So you see a CDG1 associated with the first loading event, and then a CDG1 associated with the second loading event. But both of those are much more short-lived than the corresponding MCM association. You can see that this is indeed occurring simultaneously in these 1-second frame shots of either the MCM or CDG1 fluorescence, and you can see both of them occur within a 1-second window of each other getting on the DNA. And we know this is actually true at higher time resolutions as well. So that's CDG1. Clearly we need 2 molecules. This doesn't turn out to be surprising because we know CDG1 and MCM form a complex. What about CDC6? Well, we see a very similar pattern. You can see, again, we have a first and a second MCM binding to the DNA, and each one is associated with a CDC6 molecule getting on at the same time, but coming off rapidly thereafter. The only difference, and this is consistent with our previous biochemical studies, is that CDC6 always gets on the DNA before the MCMs get on the DNA, usually by at least 10 to 15 seconds. Okay, so if I summarize this, what I would tell you is that this shows that you need that distinct CDG1 and CDC6 molecules are associated with loading the first and the second MCM complex. Now, one thing we wondered about is whether the release of these molecules might be ordered. If it was ordered, it might suggest that this is connected to the process of helicase loading, because that would suggest that there's some need to keep one molecule, CDG1, for example, after CDG6 or vice versa. And you can see already that the synchrony of these reactions is, in this case, not bad, but not great, especially if you look at the second loading. So we use this post-hoc synchronization in order to synchronize the events according to a defined event. In this case, we chose the arrival of MCM on the DNA. So whether we're measuring CDC6 release or the MCM of the CDG1 release, we measure the time of the starting point at t equals 0, so to speak, of when the MCM that they're associated with bound to the DNA. And then we ask how long until the CDG1 releases versus the CDC6 releases. And we plotted this in the form of a so-called survival curve. And this just shows what percentage of molecules are still associated with the DNA at various time points after MCM binding. And I think you can clearly see that CDC6 on average releases well before CDG1. Now this is done with 2 different experiments and comparing the results, but I'll tell you that when we label CDC6 and CDG1 in the same reaction and look at their release from individual DNA molecules, we see exactly the same result. CDC6 releases before CDG1. So this suggests that the release events are actually coupled to some important element of the reaction. So we also wondered, is this the same for the first and the second MCM molecule? So we did the same measurement, and we segregated the first MCM loading event and its associated release of CDC6 and CDG1, and the second one. And the clear outcome of this is that whether you're looking at CDC6 in blue on the left or CDG1 in red on the right, the first molecules are always released approximately in half the time that the second molecules are. And this gave us the first hint that maybe the loading of the first MCM doesn't work in exactly the same way as the second MCM. Okay? So let me summarize what I've told you so far. So we know that you start with this OCCM complex, and we end with a double hexamer, but now we know a lot of events that occur in between. We know that we had an ordered release of CDC6 and then CDC1 from the DNA, and then we have an ordered assembly of a second CDC6, a second MCM227 CDC1 complex, followed by another round of CDC6 and then CDC1 release, albeit a little slower. Now, you'll note at this point that while we've gained a lot of understanding of CDC6 and CDC1, there are two questions we haven't addressed. One of them is the key question of how you make a head-to-head double hexamer, how you load one MCM in one orientation, the second in an opposite orientation. And the second, which turns out to be related, is how many ORC molecules are required for this process. Now, let me explain why they're related. So there are two basic models to how you can load two MCMs in opposite orientations. In the top model, would suggest that you do the same reaction twice with two different ORC molecules that are bound in opposite orientations to one another. In the first reaction, use the first ORC molecule to bind, as I described before, to the C-terminal region of the MCM complexes, and then that's loaded, and then at some point you have a second ORC molecule bound, and it performs the same reaction just in the opposite orientation. These two MCM complexes can come together and form the double hexamer. A different model would suggest that there's only a single ORC molecule on the DNA that does the first round as we've described before, but that the second round is mediated not by interacting with ORC, but by interacting with the first MCM protein using this N-terminal interaction that's part of making the final double hexamer. And I should point out that the double hexamer is very tightly held together. So what I think is clear is that one key difference between these two models is the number of ORC proteins that will be present on the DNA during the loading of the double hexamer. And so Sonina did the same types of experiments I've described for CDC6 and CDC1, but using a ORC molecule and MCM label instead of either CDC6 and MCM or CDC1 and MCM. And this just shows two sample traces for those experiments. Again, you can see the first and the second MCM being loaded, but this time you see a single ORC molecule, a single increase in the ORC-associated fluorescence that lasts throughout both the first and the second loading events. And I can tell you, but won't show you, that photobleaching experiments, again, demonstrate that this is one ORC molecule, not two ORC molecules binding the DNA simultaneously. What I think you'll also notice is that while ORC stays on the DNA after the first loading event, it's rapidly released after the second loading event. And this suggests that the ORC release might be connected to completion of a double hexamer assembly. And we wondered whether this might be temporally connected to some of the interactions, release events that we looked at earlier. So we again determined the time of ORC release relative to the arrival of the second MCM. Okay, so after the first MCM, it stays on for a very long time. And what I think you can see is that it's clearly distinct from the blue line, which is the CDC6 release event, but it sits pretty close to on top of the CDC1 release events. And in fact, you can statistically show that these are not significantly different events, suggesting that the release of ORC is coupled to the release of CDC1 from the DNA. So this suggests that the one ORC model is correct. And in order to address that more, Sonina turned to something called fluorescence resonance energy transfer. And so what she did was to label one population of ORC with a green or donor one population of MCM proteins with a green or donor fluorophore, and a second population with a red or acceptor fluorophore. And it turns out you can use these two as a molecular ruler, and when the double hexamer comes together, you'll see a different level of what's called fret than you will when they're apart. So let me illustrate that here. So when they're apart, when you illuminate the green fluorophore, it will generate an emission that has the potential to activate the red fluorophore. However, if they're far apart, most of that emission will not hit the red fluorophore. In contrast, you will see what's called fret when they're within approximately 10 nanometers of one another. And then, when the green fluorophore emits, it will actually excite the red fluorophore. And so even though you're exciting only the green fluorophore, you'll still see red emissions. And in fact, the telltale sign of this is the green emissions go down while the red emissions go up when you're in a fret state. Now, importantly, when Sonina set this up, she picked sites that we knew were close together in the double hexamer to modify, and she could demonstrate whenever she saw fret, it had the hallmarks of a loaded double hexamer. So the real question we wanted to address is, when does this happen? If the model that we proposed is correct, this should be a very early event in forming the double hexamer. We don't know that this particular region will be the first place that comes together, but it should come together relatively quickly. And so we measured the time of fret formation relative to these other release events after the second MCM. Recall, we can't form fret until we have two MCMs on the DNA, so we can't do any measurements after the first one, but it also wouldn't be relevant. Okay? You can see the ORC and CDC1 coming off later than fret formation, and I should point out that the fret formation, we have an inverted axis. So as it goes down, more and more molecules have fret. Okay? So you can see in the green line on the far left, that's the earliest event we can see. In some cases, it's instantaneous upon the initial association. In other cases, it takes more than a few seconds to occur. But it's always before CDC6, and it's always before CDC1 release, consistent with the model with this being a very early event in the recruitment of the second MCM molecule. Okay, so let me summarize what I've told you so far. Our original understanding was this initial association event where we had ORC, then CDC6, then MCM and CDC1, and we formed the OCCM, and then there was a lot of question marks, and we only knew there was ATP hydrolysis. Now we have a much better understanding. We know the first event after the OCCM is the ordered release of CDC6 and CDC1. CDC1. And I'll tell you, but didn't show you that we know the ring is in fact closed at that point. Then we have a second CDC6, a second MCM CDC1 binding, and this leads to the formation of the first double hexamer interactions, and we believe that's mediated by the N-terminal interactions between MCMs, and that's in turn followed by the release of CDC6, and we believe the simultaneous release of CDC1 and ORC from the DNA. And there's several properties of this model that we like in terms of driving the formation of a double hexamer of DNA, a double hexamer of MCMs on the DNA. So one is that because the ORC stays bound after the first MCM is loaded and then is only released after the second MCM is loaded, you will drive the reaction towards double hexamer formation rather than having the possibility of having single hexamer interactions. And secondly, I hope it's obvious by now, by using the N-terminal regions of the two MCMs to recruit the second molecule, you will ensure that you always form the head-to-head double hexamer at the origin. Clearly, there's much more that can be done using single-molecule studies to understand the events of replication initiation. For example, we can start incorporating mutant proteins into this process to understand when key events such as HD hydrolysis or key interactions occur and how they influence this process, especially kinetically. In addition, we're very excited to use FRET to monitor additional protein-protein interactions. I showed you using it to monitor the formation of the double hexamer, but you could also use it, for example, to monitor the opening and closing of the MCM267 ring during the loading process, which would be one of the most key events in understanding loading. Finally, much of the replication process has been reconstituted in purified proteins, and so extending the same approach to look at the helipase activation and ribosomal assembly process is a key goal for my laboratory. So I'd like to end by acknowledging our collaborators. As I said at the start, all of this work was done in close collaboration with Jeff Gellison's lab, as well as Larry Friedman, and they, as I said, are integral to the analysis of all the data, as well as building microscopes that allow this kind of research to be done. I'd also like to thank the latest lab at the Janelia Research Campus, who developed a number of long-lasting fluorophores or photostable fluorophores that were essential for the success of this research. And finally, I want to thank the members of my lab. Most of the work was done by Samina, however, she was assisted by Nikolai Becca, who labeled many of the proteins, which can be one of the most challenging parts of these studies. And finally, I just want to thank the funding sources, the National Institutes of General Medical Sciences, as well as the Howard Hughes Medical Institute. Hello, my name is David Alice. I'm a professor and head of laboratory at Rockefeller University in New York. My laboratory studies chromatin biology and epigenetics. If you were able to watch my first My Biology talk in this series, I was trying to introduce what is epigenetics and give some of the historical perspective, some of the big experiments that led to the concept that there may be something more to our genome, and that led to our concept of epigenetics. Today, I hope I can build on sort of some of those thoughts and tell you about how does epigenetics impact some of the development in disease, focusing a little more on some of the more current experiments that are ongoing in my laboratory and others. So again, the same sort of orientation would be epigenetics as a term, something in addition to our DNA, on top of DNA. It traces back to Conrad Waddington, who in the 1940s coined the term to try to explain how in development would a young embryo sort of ultimately pass through development and give rise to many different cell types, whether it's blood, nerve, skin, whatsoever. And once those sort of cell identities have been established, all from the same genome, then the question would be, how could that happen with the same genome? So he was fascinated by that process. Of course, once the differentiated cells have been established, in general, they know who they are and what their disease. So blood cells will give rise to other blood cells and so on. But every once in a while, so there's a memory aspect to it. But every once in a while, unfortunately, some of these epigenetic landscapes get confused and give rise to sort of cells going backwards in development, where they become more undifferentiated, more proliferative, and the outcome of that often is a cancer of some type. And I'll talk a little bit more in my lecture of some of what we're doing that has sort of a cancer connotation. So to give you some insights, I have to explain how the genome is packaged. If you look at this cartoon of a eukaryotic nucleus, you can see that although we're depicting some naked DNA, in fact, very little of our genome is naked Watson and Crick DNA. There's a fundamental repeating spool, if you will, that you can see in the lower right-hand corner is now at atomic resolution, thanks to Tim Richmond and Caroline Luger. That fundamental repeating unit, like a bead on a string, is called the nucleosome particle. And if you follow up on the cartoon, we don't understand quite in the field all the details of how these nucleosomes pack and pack and pack to give rise to higher order structures. But ultimately, 2 meters worth of DNA have to package into a eukaryotic nucleus. And I think it's intuitively obvious that there's going to be regions of the genome that are reasonably inaccessible, and there's going to be regions of the genome that are accessible. So let's take a look at the nucleosome in even higher order magnification. So I think this is a very elegant structure. This was a landmark structure in 1997. And I just use this cartoon to sort of ask you to notice that emanating outside of the DNA are histone, what we refer to in the red box as tail domains. And you might be tempted to think, well, they can't be important. They're not really packaging the DNA. And in fact, you can set up situations in the lab where you can chop the tails off with either enzymes or with genetics. And indeed, you still make a very good nucleosome particle. So why has nature held on to the tails being as conserved as the rest of the histones? Where in fact, if we pick on the blue histone, which is sort of going to be the star of my show, histone 3, The tail of histone 3 is conserved essentially from yeast to man. So if it doesn't package the DNA, why are these tails being held so tightly in evolution? Well, the answer will be that those tails are a very attractive landing pattern for covalent modification. And engage the histone chromatin fiber that way. And so I think we now have lots of insights from many laboratories of molecules that fall into these categories, in the lighter category, the eraser category, or the reader category, and many of these have become effective drug targets and proven to be quite useful in clinical settings, in part because the DNA is not altered. So this may not be fair to call this a reader, but there would be a very powerful cell biological tool that I think my laboratory and others have put to good use, and that would be, can you find an antibody molecule, can you generate an antibody molecule that might be highly specific for the modification of the post-translational modification on the tail domain. I want to give you some spectacular views of where these type of antibodies started to let the field know that there's a lot going on here with this epigenetic regulation. This is a classic cover of a Cell article that was published in 1993 by Brian Turner and his colleagues, where they generated an antibody I'll refer to as anti-acetylated, so that would be the chemical modification, anti-acetylated H4. And they spread mammalian chromosomes out on the slide. I think you can see on the left there's a lot of kind of green staining chromosomes. And they made note with a counter stain that was enabling them to see sort of the underlying non-antibody stain, that one chromosome in the dashed box with the circle, arrow pointing to it, that there was one chromosome that was lacking the green stain, the yellow-green stain. And that, in fact, turned out to be the X chromosome. And I have to tell you for completeness, these were female mammalian cells. So they were now exhibiting a phenomenon called X inactivation. This is one way the two X chromosomes in a female will be kept sort of equivalent genetically to the single X chromosome in the male. And here's a higher mag, just so you can see how really spectacular this was. This is the same image, just blown up. So you can see it's really quite clear that inactive X chromosome labeled XI is really not staining with this antibody. This is an assay we refer to as immunofluorescence. And I think if you look carefully, some of you may notice that some of even the chromosomes that are largely yellow staining have little zones where you see the underlying red. And in fact, those are regions where we don't have much acetylation. And of course, the correlation here is that when you don't add these acetyl groups, in general, these genes, and in this case, a whole chromosome, are not transcriptionally active. So hypoacetylation attracts gene inactivity. Now you might say, well, maybe every methyl or acetyl mark is created equal. Well, here's the real sort of amino acid sequence across the top in blue of the histone 3 tail. It's not the entire tail. This is in one-letter amino acid code. But I'll just ask you to look at two lysines. I should say that's where these chemical groups are often added, a lysine residue, that's K. And so you can see if you generated an antibody to K9, you would be able to sort of potentially get an antibody that might read the red sphere, if you will, a methyl mark. And I'll just tell you that that's embedded in the sequence, too much detail, ARKS. Now you can see if you expand down and look further into the histone 3 tail, skipping some residues, you'll see another ARKS motif in the yellow box. Again, that's a lysine at 27 now, so we're further into the tail. It was known to be methylated. We could generate an antibody, me and others, to the lysine 27 methyl. And I need to sort of remind you of a silencing paradigm that came up a little bit in my first biology lecture. When you want to silence genes and use methylation, you ultimately have to remove the pre-existing acetyl marks with histone deacetylases or HDACs. That sets the lysine up where it can now obtain a methyl mark by histone methyltransferase, a writer. And then there are reader proteins, X and Y, that are being dictated here that read these methyl marks. So the question is a K9 methyl mark identical to a K27 mark? And here's some real, again, immunofluorescence staining patterns. This takes advantage of the Drosophila has a beautiful set of chromosomes. They're called giant polytene chromosomes. All you need to know is that they're big and they're huge. And you can spread these chromosomes out on a microscope slide. And on the top two panels shown in red, you can see the staining with either a K9 methyl antibody on the left or a K27 methyl antibody on the right. And I hope you might agree that they don't look at all the same. And in fact, what we know is the sort of genetic epigenome area on the left, where there's like a central blob of red staining, those genes, that genome sequences, are really heterochromatic and always off. In contrast, the upper right red panel with the K27 methyl staining is genes that are known to be out on the chromosome arms, that should be clear, and they're regulated off. They're not always off, they're regulated off. And below each panel are antibodies that were developed to the corresponding readers. It's too much detail, so on the lower left, we'll just consider it X. On the lower right, we'll just consider it Y. But I think you can see there's a pretty good agreement of the reader sort of tracks and co-localizes with its mark in either of the two K9 versus K27 situations. So this is sort of like a way to look at the genome in Drosophila. Now, yeast is an organism I want to set up carefully later in my talk, but I want to say, but are all methyl marks silencing in nature? No, they're not. And in fact, if you look at the very tip of the H3 tail, lysine 4, that was shown by many laboratories to be an on mark. So not all methyl marks are silencing, some are activating. But yeast as an organism has this amazing property that you can obtain genetic knockout strains, and not all of them will kill the organism, so you can get viable yeast strains that have knocked out individual genes. And all you need to know for this experiment is the methyltransferases, the writers that add methyl marks, they all have a domain called a set domain. That's too much detail. But then once the yeast genome was sequenced, we could ask, well, how many set domains are there in yeast? And the answer was 7. And so you could just order knockout strains for each of the individual